Of some of the information that we covered in class for our first protein modeling day. Now, in terms of our building blocks, we have amino acids that are used to build proteins. So we're talking about Legos becoming things like castles or TIE fighters or other large Lego kits that you might have played with growing up, or the same idea as building a pizza out of lots of different toppings. Each pizza and each Lego kit will have a different unique structure based on the things that build it up, and that will then determine their function. So when we talk about our building blocks for amino acids, we need to talk about three key components. The first is the amine group, which is our left-handed side of our molecule. Every amino acid has the same exact NH2 structure with two positively charged hydrogens. Secondly, we have a carboxyl group on the right-hand side. This is, again, a shared feature amongst all 20 amino acids where we have a C double bonded to a negative oxygen and then an OH group here as well. This is our acidic part of our molecule. Over here is our nitrogenous base. These two molecules form the backbone of our protein, or what are your lines in your modeling. And then finally, we have our R group, and our R group is the thing that makes each pizza different or each taco different. It is the special sauce or the ingredient that distinguishes it from all of the other pizzas. We have four types of R groups. We have hydrophobic or hydrophilic, positively or negatively charged molecules that go here. Of the 20 different amino acids, there are 20 different structures that make them have one of those four characteristics. So you should have rolled out 20 M&Ms that gave you a random order of hydrophobic, hydrophilic, negatively and positively charged amino acids, and you have 20 different options really in there. You might have four colors, but any of those colors could be 1 out of 5, 1 out of 7, 1 out of 5, different types of amino acids that make up the 20 that exist in the human body. So then we talked about the protein primary structure, the primary structure of proteins, which is just our beads on a string. We add amino acids to each other via a peptide bond, which you can see right here. An oxygen and a hydrogen fuse with a hydrogen from amino acids amine group, forming a water. This is why it's called dehydration synthesis. OH plus H is H2O. And then we have these two amino acids bonded. This forms in the ribosomes when RNA is read. Remember, the more amino acids that you have, the more possibilities that there are. We don't have any shape or flavor or characteristic yet, but remember that in our amino acid, we have a positively charged NH2, and we have a COH, or acidic carboxyl group that's negatively charged, plus our R group. So let's go take a look at my drawing and think about what we're doing here. I have 4 amino acids, 2 times, that represent the first 2 curves of our secondary structure. I'm right now just going to give them specific flavors. I'm going to make red hydrophobic. So I'm just going to do 2 hydrophobic molecules to start off our adventure. I'm going to make our third amino acid green, which I'm going to say is positively charged. I hope I remember that later. I'm going to make our fourth amino acid yellow, which is negatively charged. I'm going to keep 1 and 2 here, blue, making them hydrophobic, or hydrophilic, I'm sorry. And I'm going to make 3 and 4 like charges and make them green. So what we have right here is our primary structure, which is just a string of amino acids forming a peptide bond background, or backbone. And we don't have this curve yet. So let's explain how we get to this alpha helix curve. We know that amino acids have negative and positive sides. So when we put them all in a peptide bond line, those interactions between positive and negative groups start to come into play. And what happens is every fourth amino acid down or upstream of an amino acid becomes attracted to those other two. So in a way, we have four cardinal directions. We have amino acids on the facing side, the opposite side, and the two peripheries that form these same interactions. And we're going to see how that looks in a minute thinking about circles. The alpha helix is symmetrical, and every twist, every bend from this point to this point contains 1, 2, 3, and 4 amino acids. 4 amino acids fit on each coil of our alpha helix. So if I look at my drawing, I now have 1, 2, 3, 4 different amino acids on the same coil, and then it repeats again. I have my first one here at what would be considered the west side. 2 is at the north position. We're spiraling around. Imagine you're looking at this coil from the bird's eye view. We have our third one here, and then we finally finish the coil with our fourth one, which is our yellow negatively charged. I then have my first coil at the western position here. I have my second amino acid here at north. Some copying and pasting issues I see. And again, we're just thinking about what would it look like if you looked down the center of a slinky. You'd see a circle. And so on and so forth. So every alpha helix contains 4 amino acids. And the reason why is their negative and positive regions are attracting. So what that looks like, if I want to think about a line, is this molecule is attracted to this molecule based on negative and positive interactions. Let's make that big, bold, and orange. At the same time, the same amount of attraction is occurring down here with our 2 first amino acids. This explains why the alpha helix is so symmetrical. We have 4 sides with equivalent positive and negative interactions between our different amino acids. We're not yet thinking about hydrophobic or hydrophilic. We're just going back to that idea that each amino acid has positive and negative sides. And if the coil occurs, it's occurring because there's 4 symmetrical sides of pairs of amino acids feeling the negatives and positive attractions. So then we can start playing around with that symmetry when we get to our tertiary structure, or where we start to see coils and bends occur. And this is where we want to think about some just, abstract ideas with slinkies. So when we build our tertiary structure, which is where we start to see actual bends and folds in our proteins, this is where the R groups come into play. Hydrophobic amino acids are going to fold inwards. Positively and negatively charged molecules are going to come together. And then finally, neighboring charges are going to repel each other. Hydrophilic regions are generally going to fold outwards. So let's go take a look at my drawing. And I think it might be easier to think about what's going to happen with our different circles. So I'm just going to be using arrows to show you what I think would be happening here. My first 2 coils, or my first 2 amino acids of my first coil, are going to be bending inwards. And the reason why is they are both hydrophobic. I'm going to think about what that would look like here below as well. Excuse me. By thinking about this part of the circle contracting inwards and shrinking inwards. At the same time, 3 and 4 want to come together, so I might imagine these 2 molecules bending in towards each other. If 1 and 2 here wanted to fold inwards and shrink into the center, the opposite is going to be true for 1 and 2 for my second coil. The circle here is going to get bigger as a result of these two molecules wanting to spread outwards. I could imagine this part of the coil opening up, getting wider. The U opens up more, it's less steep and it's wider. Over here, the upside down U gets smaller and steeper. And then finally, 3 and 4 are going to want to repel each other because they are like charges. So I could imagine this part of the circle getting even bigger and wider and straighter because 3 and 4 want to get apart from each other. So as you are drawing your tertiary structure, you're taking the symmetrical alpha helix, which is due to just pairings of amino acids attracted to each other, and you're contracting, opening up, and shifting these hills as a result of R group interactions. That is all I need you to know for today in terms of the primary, the secondary, and the tertiary structure. In terms of the modeling questions that you see below the directions for folding your protein, I expect you to be able to answer the first 3. We'll talk about the 4th one tomorrow in class. I hope this video has been helpful. I know that this topic is confusing and difficult, so I appreciate your patience, your questions, and you asking for support. Have a good rest of your day, and I will see you tomorrow or soon.